So now we're going to look at the 3 steps involved in a cell signal, the cell receiving a signal, the signal being passed from the cell membrane into the cell in a pathway, and then the way that the cell responds. Again, chapter 11 goes into a lot more detail on each of these steps than we're going to, so please use the chapter outline that I posted or this PowerPoint as a guide for what things I'm going to ask on the test, and if it's not included, you can assume that that's not going to be on the test. So in order for a cell to receive a signal and then have some kind of response to that signal, it's going to follow these 3 steps. First, the signal has to actually be received. So this is called reception. The reception of a signal is almost always going to happen at the plasma membrane. There are some exceptions to that, of course. So the plasma membrane has this transmembrane protein here that is a receptor. That receptor's got some sort of receiving space right there. And then here's my signal molecule. Oftentimes the signal molecule is called a ligand. The word ligate means to bind. So a ligand is going to bind into this protein, and that physical connection, the fact that this little ice cream cone fits in this triangle spot right there, is how it receives the signal. If a cell doesn't have the proper receptor, it's not going to receive that signal. Once the signal is received, it needs to be transmitted. So this is the process of transduction. Remember, trans just means across. So this is how we're going to get the signal from wherever we received it to wherever we need it to go. And this is going to occur in a series of steps. So you can see that the receptor is going to send a signal to enzyme number 1. Enzyme number 1 sends a signal to enzyme number 2. Number 2 sends a signal to enzyme number 3. 3 is a fairly short pathway. I would expect to see many more steps involved in this as well. And as each of these steps occur, the signal is moving physically closer to whatever response it's going to cause in the cell. The last step of cell communication is the targeted response. So each specific signal is going to target a specific response, and that response is going to be different depending on what the signal is, what type of cell is receiving it, maybe the environmental conditions inside of the cell. And so eventually the last step of the pathway is to send the signal to, sometimes this is the nucleus, and it's going to signal for the cell to start replicating or to turn on and activate a specific gene to make a specific protein. It may even signal to the cell that it's programmed for death. So there's lots of different types of cellular responses to signals. All right, so now we're going to start with step 1, and we're going to look at some different types of receptors. The relationship between a ligand, which is my signal, and the receptor is extremely specific. So if my receptor protein is shaped like this, it's not going to bind a ligand in a circle shape. It's only going to bind a ligand that actually fits into that specific space. So this is very, very specific, just like enzymes. When this happens, and this actually binds into the receptor, oftentimes this receptor is going to change shape. So it might have a bulge that comes out here, or it might just have a slight change of shape on the other side of the cell membrane, which is going to go ahead and kick off the next step. The vast majority of these, again, are going to be on the membrane. There are a few exceptions that are going to be found inside of the cell. So now what we're going to do is go through several different types of receptors. So for each of these receptors, I would highly recommend making some kind of flashcard so that you can remember the receptors in case there's a multiple choice question or something about them. So the first and most common type of receptor is called a G protein coupled receptor. So I know this is a long term. Let's look at the words. Receptor just means I'm going to receive. A couple just means 2, right? There's 2 things together. And then this is the thing we probably don't know here, which is a G protein. And a G protein is just a specific type of protein that exists inside of the cell membrane. So here is my G protein. G proteins, instead of using ATP, which is adenosine triphosphate, they use GTP, which is guanine triphosphate. So it's the same basic structure with 3 phosphates, but it's got a slightly different nitrogenous base, so it works fairly similar. And that's part of the reason these are called G proteins, is because they use GTP instead of ATP as their energy source. So a G protein is coupled or paired with a membrane receptor. So you can see in this image, here's my receptor here. It's got a space where it can receive a signal, and my G protein is right next to it. It's not physically contacted. This G protein is inactive. When the receptor receives the signal, so the ice cream cone fits into that spot, what happens is it changes the shape of the receptor, which is going to trigger the G protein to go ahead and use a GTP. That GTP is going to change the shape of the G protein, which is going to cause it to move inside of that membrane, and then it's going to activate some sort of next step. So this next step might be the first step in the transduction pathway. So this is a very generic G protein image, but G protein is the largest family of cell surface receptors. So these are the most varied, they have the most different pathways that they're associated, and the most, these are the beginnings of a very large group of different signal pathways. Another type of receptor is called a receptor tyrosine kinase. So let's talk about this word here, kinase. ASE means that it is an enzyme. So an enzyme is going to be a protein that is going to catalyze a reaction. A kinase is a specific type of enzyme that adds phosphates. So a kinase is going to take a phosphate and stick it on another molecule. Oftentimes, moving a phosphate is just enough to change the shape of a molecule and activate it or turn it on and trigger a response. So these are typically going to use ATP, and the kinase is going to take that ATP and give it to some other protein. These can often trigger multiple pathways at once, so it's not always going to be a linear path through the cell. A single receptor might be able to trigger 2 or more different pathways. The key characteristic of these that's very different is that they form a dimer. So here you can see there's kind of 2 halves here. Here's one receptor tyrosine kinase, here's another one, and they are separated here by physical space. Once those both bind to the signal molecule, so it actually requires 2 signals here, they're going to come together and form this dimer. So remember, di means 2, so that's just 2 molecules stuck together. Once the dimer is formed, then the kinase thing happens, and they're going to pull phosphate from ATP and stick it onto the tyrosines. And now that the tyrosines have their phosphates, their shape has been changed, and it's able to go ahead and activate some kind of cellular response. And so in this image, you're seeing that it's got one pathway that's going to go this way, and another pathway that's going to go that way, and probably end up with 2 different cellular responses to this one signal. These receptors are misfunctioning or they're mutated. These can often be associated with different types of cancers and cell cycle regulation. Another type of receptor is called the ion channel. And so we already learned about ion channels in passive transport. So this is going to be just a membrane protein that's going to function in facilitated diffusion. So a ligand-gated ion channel. So the ion channel is going to allow a specific ion to move through it. So if this is a sodium channel, then it's going to allow sodium to move through it. This gate here is just like the door. It opens when a ligand binds. So think of it like a door that requires a key. So whenever the ligand, which in this case is probably sodium, or it could be a different signal molecule, whenever that signal molecule binds to the receptor, what's going to happen is it's going to open this gate, and then once the gate is open, the sodium is going to be allowed to move. It is able to close again, so if you were to remove the ligand, it would change its shape back to its original closed shape, and then that channel would be closed. So here's an example. Here is an ion channel. Let's say that this is calcium. So some signal molecule, we've got our ice cream cone here, is going to fit into here. And once it does, it's going to cause this receptor to change shape and open this channel. And once the channel's open, the protein doesn't actually have to do anything. The calcium can just move from an area of high concentration to an area of low concentration until it reaches dynamic equilibrium, or until this ligand is removed, and once that's removed, it will close up again. And so the thing that causes the cascade now is this influx of ions is now going to trigger some type of pathway or cellular response. Here's our last type of receptor, and this is an exception to the rule of having to be on the membrane. So intra means inside. So intracellular means it's inside the cell. So an intracellular receptor is going to be found either in the cytoplasm or in the nucleus. It's pretty much going to be anywhere inside of the plasma membrane. And these are going to be either small or hydrophobic chemicals that are able to easily cross the plasma membrane just using simple diffusion. So in this example, we've got aldosterone, which is a steroid hormone, very similar to testosterone or estrogen in shape. And because it's a steroid, it is nonpolar, and thus it is easily able to pass that plasma membrane. It's not very large in particular. And then what happens is it's able to find its receptor protein inside of the cell, which is going to go ahead and trigger some kind of cellular response, in this case, turning on a gene to eventually make some new protein. The second step in the signal pathway is transduction. That's how do we get the information from where we received it at the receptor in step 1 to the target in step 3, which is going to give us our response. This is often a chain of interactions. So this is going to be many individual steps where the first step causes the second step, and so on and so on and so on. So we're going to see we activate one protein, which activates another protein, which will eventually reach a response. At each step, the signal is changed into a different form. So the different form might be in the shape of a secondary messenger. It might be in the form of changing a protein's shape into a different shape. So at every step, there's some change that takes place, and then that change is going to cause the next step in the pathway to occur. One of the most important reasons for having these kind of complicated transduction pathways is that they can help amplify a signal. The word here, amplify, is going to mean to make it bigger. Right? Think about an amp to amplify music. It's going to take the sound that you put into the amp and make it louder. So this is a way where cells can receive a very small signal from the blood and turn it into a very big response without needing lots and lots of signal molecules to get a big response. So most transduction pathways are going to involve what are called secondary messengers, and these are just messengers that occur inside of the cell. So the most common is cyclic AMP or CAMP. So in this image, you can see that we've got a G protein coupled receptor here. When it receives its signal, so here is my signal reception, step number 1. The G protein is the second step, and it's going to signal an adenylyl cyclase here, which again, you don't have to remember these enzyme names. Adenylyl cyclase is going to cause ATP to give a phosphate to CAM, which is going to turn it into CAMP. And this is my secondary messenger. The CAMP is going to go find a proper protein kinase. Again, kinase is going to add phosphates to things. And this kinase is going to add a phosphate to something, change its shape, and then cause additional steps. So this is just one part of the transduction pathway. Just like G protein coupled receptors are the most common and varied receptor, cyclic AMP is one of the most common and widely used secondary messengers within the cell. So it might seem a little bit overly complicated for cells to have 15 different steps that they need to go through in order to get a signal from the plasma membrane to the nucleus. Why not just send CAMP directly to where it needs to go? And part of that is because each step allows us a chance to increase the response. So here's an example. I've got one G protein. So this G protein is going to receive one ligand, right? Or this is the signal. So this means that in the blood, I don't actually need to have a lot of hormones or whatever signal molecule flooding the blood. I can have a few. So one is going to bind to the cell. So this one G protein is going to activate 100, 10 squared is 100 adenylyl cyclase, this is now the second step. Each adenylyl cyclase will activate 100 CAMP. So now we go from 100 adenylyl cyclase to 1,000, 10 to the fourth is 10,000 cyclic AMPs. So we started with one ligand and we're already up to 10,000 secondary messengers. Those secondary messengers activate one protein kinase each, so we're still at 10,000 protein kinase A. Each of those is able to activate 10 of another phosphorylase kinase, so 10 to the fifth is now 100,000 phosphorylase kinase. And then eventually it's going to reach its final step, which is to activate glycogen phosphorylase. Each one of those is going to activate another 10, so we end up with 10 to the 6, which is 1,000,000 active glycogen phosphorylases. And again, you don't have to remember the name of these molecules. The point here is that we started with one signal coming from the blood, getting to the cell. And we ended up with 1,000,000 molecules involved inside of the cell in the response. And so this is amplification, going from 1 to 1,000,000, and we only increased it in 6 steps. Having 6 steps here allows us to drastically increase the response that a cell can have to even a very small signal. And eventually we will get to the third step, which is the cellular response. So we're using response here as kind of a generic term. The response is just whatever the cell does in reply to that signal. So up here we've got reception, transduction, and then in this case our response here is happening in the nucleus. And what it has done is it turned on a transcription factor, and here's my DNA, and it's going to go ahead and start protein synthesis to make some protein product. So this can happen in the nucleus. A response can happen in the cytoplasm. Sometimes this is going to turn a gene on. It can turn a gene off. It can create enzymes. It can make other proteins. Sometimes this is going to go ahead and lead to other cellular activities like cell replication. So if we had a growth factor, right, this is a growth factor, this is maybe going to go ahead and stimulate that cell to replicate, or a different signal may even signal that that cell is mutated or damaged and that it needs to go ahead and be demolished. One of the other advantages to having lots of steps in the signal pathway is that there's lots of places for variation in responses. So the specificity of the response, so what response you get from each signal, is specific to each cell and specific to each pathway. So here we've got cell A, and this is cell B. They're going to receive the same signal. In cell A, that signal has one pathway and it proceeds to response number 1. In cell B, that same signal has a different pathway that actually splits into 2 responses, and we get 2 completely different responses from the same signal, just because these 2 different cells have a different transduction pathway, right? That's step number 2. Sometimes along these transduction pathways, so on the way to the response, you will meet what's called a scaffolding protein. So if you think about a scaffold like a building that's under construction, the scaffold is the thing that the construction workers build on the outside, and it looks kind of like bars and they climb up it to be able to do work on the building. A scaffold is going to be kind of on the outside, and it's holding these 3 proteins together. And what's happening is these are maybe 1, 2, and 3 steps in that transduction pathway. So when the signal is received up here at step 1, that signal can go to the scaffold process, and then it can go through that transduction pathway fairly quickly. And so that's going to make transduction really efficient. And it could be just efficient because the three molecules are physically right next to each other, and so the pathway can go quicker. So oftentimes these scaffold proteins are found in pathways where the transduction is going to be very efficient and quick. So up to now, we've been talking about how to start a signal and how to send the signal. But at some point at the end of the message has been received and we turn it off. So eventually the signal has to stop. And there are several different inactivation mechanisms, and these are important for cell functioning. You don't want to turn a gene on and then just leave it on. It's not like you don't turn a light on and just leave it on. Eventually you do want to turn it off. The easiest way to do this is stop sending the receptor. So when there's fewer receptors, they're not going to bind. And when they don't bind to the receptor, then the signal will turn off. The receptor will go back to its inactive state, and then the entire signal pathway will stop. So it'll unbind and revert to being off. This can also happen in the relay, so that transduction pathway. Once the receiver is off, then those relay molecules get turned off as well, and they will go back to their inactive forms, and then eventually the response that the cell was giving will turn off as well. And then it can turn back on again as soon as that signal is received again. So we've talked a little bit in general about the different types of signal responses that cells can give. And I want to talk about apoptosis just a little bit here. So say this to yourself, apoptosis. It's a fun word. And what this means is programmed cell death. So this is when a cell is killed specifically because it needs to be killed or it's time. So what happens is in this cell, the components inside of the cell are chopped up and put into vesicles. And those vesicles form kind of lobes, and the scientific term for that is blebs. And so over here you can see a cell with blebs. And this is kind of like an internal demolition. So what's happening inside is the lysosomes inside of those vesicles are chopping up all of the packaged pieces. But instead of just popping the cell and letting them all out, it's doing it in a way that's contained. So it stops it from leaking out because it might damage the neighboring cell. And so this way, only this cell dies, and this cell is able to maintain its proper function because this cell is going to be demolished internally and not leak out or damage its neighbor. Apoptosis is common. Everybody has done this, and this is a normal part of life, is killing off cells that are damaged or cancerous or just are part of proper development. So if you look at this down here, this is an embryological hand between about 7 to 9 weeks of development after fertilization. So a baby's hand starts out as a nub, and you can kind of see that each of these little bumps is going to be a digit, and there's membranes in between all of the fingers. So eventually, the cells between the digits start to go through apoptosis because we've evolved to have very dexterous and flexible fingers, and we don't actually have membranes between them. Eventually, in a week or two inside of the womb, this actually happens fairly quickly, the spaces between the digits are clearly defined and none of that webbing remains. So if you know anybody who has webbed fingers or toes, that's just because their cells didn't properly go through apoptosis while they were a fetus. Apoptosis is sometimes used by tumor suppressor cells to contribute to killing cancer. And so if apoptosis is stopped or it's not properly brought about, then that's how some cancers are formed. So a cancer would be a cell dividing when it's not supposed to. And if that cell was supposed to be killed and then wasn't killed, it could continue to go on and form some form of cancer.